Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Michael Shadlin visiting us from the University of Washington. Michael is Associate Professor of Physiology and Biophysics and part of the staff of the Regional Primate Research Center at UW. He did his BA degree in biology as well as his MD at Brown University. He left after a couple of years on a temporary leave, doing something very exotic for a medical student, and headed off to UC Berkeley to work on his PhD degree in stereopsis perception. And then after he finished that, came back to finish up his MD degree. And he actually is, believe it or not, a practicing neurologist as well as a basic research scientist these days at UW, spending about a month a year at the Veterans Administration Hospital seeing patients. Today he'll be talking about the neurophysiology of decision-making, rate differences, and log likelihood ratios. Well, it sounds like a very interesting topic at the cusp of biology and probability.
Well, thanks very much for the invitation. It's the first time I've been to Microsoft. It's very exciting. And I take it that everyone can hear me fine? Yeah, okay. All right. Well, in the Second World War, the German military encrypted their communications using this device called the Enigma machine. And what it did was it converted a letter in an original message to a new letter, of course, and at the same time advanced a series of rotors so that the mapping changed every time a letter was entered. And the Germans thought that this code was unbreakable because of the enormous number of possible mappings between original letter and encoded letter. But this guy over here, who you all recognize as Alan Turing, found a way to break the Enigma code. And it turns out that what he did was he found a way to analyze sequentially the letters in pairs of messages and make decisions, simple decisions, binary decisions about the state of the Enigma machine. And importantly, he developed a way to accumulate evidence for and against hypotheses and terminate the decision process when sufficient evidence was reached. And in effect, he had invented secretly, at the time, sequential analysis. And at the end of the talk, I'll tell you how he did this. It turns out, though, that the trick that Turing developed is similar to the way our own brains, here's a brain of a monkey, the way our own brains decipher, if you will, the coded messages in the sensory cortex. And I'm going to tell you today some things about how the neurons in our brains make decisions. So here's an overview of the talk. I'm going to talk about, just give you some background. Since you're not biologists, I'll tell you how neurons in the brain underlie a very simple decision about motion, and we'll try to understand both the accuracy and the speed of those decisions. And I'll try to make a connection to Bayesian decisions and sequential analysis. That'll bring us back to Turing. And finally, I'd like to posit, I think, as a point of sort of fun discussion, what I call an intentional framework for information processing in the brain. Here's some things I'm not going to talk about since we don't know each other. I thought I'd at least sort of mention some things. You can either ask me questions about it at the end or not. So there's some experiments we've done where we've manipulated prior probability, and maybe when I'm hearing more about what you guys are interested in, I might want to talk about this in more detail at the end. It's more complex decisions than the ones I'm telling you about, and then I've published a fair amount of papers on basically the neural code of information in the brain, and in another line of work, how brains keep track of time. So I'm just laying that out for you so that if you want to ask me questions about it, you can. All right, so what's a decision? So when President Bush decided to invade Iraq or not, he made a decision. And a decision consists of some combination of evidence from the world, good or bad, some prior knowledge, prior probabilities, and some estimate of the potential costs and benefits of the decision. And most decisions involve some degree of chance. And so we need to come up with a framework that allows us to make computations in the brain using a common currency for evidence, prior probabilities, cost, expectations. And we have to deal with this, the idea that information is not often perfectly reliable.
That's not like airbrushed in the 8-ball there.
I have no idea, actually. I got this off the web. So Okay, so here's the decision task that we use in the lab. It's a lot less dangerous than the one that the president decided to engage in. And probably much more ethical, too. So here's a monkey staring at a computer screen. He sees this little spot, and then he sees two targets that appear in his peripheral vision. These are going to be choice targets that the monkey's going to use. He doesn't talk to us, obviously, so he's going to make eye movements to tell us what he sees. And what he sees is a pattern of random dots. They're dynamic. Most of the dots are just flitting about randomly on this screen, but some small fraction of them are moving either to the right or the left, that is, towards this target or towards this target. And the monkey's job is to decide the direction of motion. Here's what he sees. So he'd be looking down here at this fixation cross, and then he's trying to decide if the net direction of motion is to the right or the left. And this is easy. You should all be seeing rightward motion here, and if you can't, then I'll practice as a neurologist at the end and see you. Okay, so now the monkey in these tasks makes his decision, and as soon as he's ready, which would have been an eternity ago now, he would break fixation and move his eyes, and we would sense his eye position and turn off the random dot motion. And here's the monkey telling us he thought the motion was to the right, and if he's correct, he gets a reward. And so that's the basic behavioral paradigm. We do this with humans. We do this with monkeys. They're pretty similar. Okay? Now, you get two behavioral measurements from this kind of a task. You can measure the monkey's accuracy and the speed of his decisions. Okay, so here's the probability that the monkey identifies the direction correctly as a function of motion strength. So this motion strength refers to the percent coherence. This is really basically the probability that a dot will be replotted in motion as opposed to randomly on the screen. So when the motion is strong, the monkey's perfect, and I really do mean perfect. And when we make the motion weaker, the monkey's performance progressively falls off towards chance, which in this case would be 50% correct. Okay, now if you ask how long it takes the monkey to make his decision, we can now plot the reaction time. So this is the amount of time that elapses between onset of the random dot motion and the beginning of the monkey's eye movement response. So again, if we look over here, when the motion's strong, it doesn't take him very long at all to finish the task, about on the order of 400 milliseconds. But as we make the motion progressively more difficult for him, we extend the reaction time out to over 800 milliseconds. Now, not all of the reaction time is spent deciding. There are sensory and motor latencies that have to be incorporated into this total reaction time between onset of random dot motion and onset of the eye movement. But if you think about it, there's at least 400 milliseconds that separate the easiest from the hardest stimulus. And it's taking some time for the monkey to decide even about the easiest stimulus. So we have in our hands something like 400 plus milliseconds of decision-making time. And from the point of view of a neurophysiologist, that's an eternity. It allows us to go in the brain and record neural activity while the monkey is deciding, but before he's finished the decision process.
How did you instruct the monkey to make the quick decision as quickly as possible?
Well, that is a serious trick. So first of all, I should say that training monkeys to do simple tasks like associate eye movements to targets with instructions coming in the form of random dot motion, that's pretty easy. A few weeks maybe. Okay, a few days sometimes if you're lucky with a smart monkey. But getting the monkey to do threshold psychophysics is another story. Getting the monkey to do threshold psychophysics at a variety of geometries, because we don't know what neuron we're going to encounter, so different places in the visual field, different directions, and so forth, that adds more time. And getting him to do stable speed accuracy is another story. And it turns out that, let's come back in a while, and I'll tell you why I think the monkeys get the particular speed accuracy trade-off that they achieve. Okay? It turns out to maximize their reward per unit time.
If the monkey's very hungry, do you get a shift of that curve?
Well, the monkeys are motivated by, we control their water intake. So in the morning, they don't get their water. And so when they come out of the cage, then they go and they kind of work for water. Every time they get something right, they get water or juice. And so when they're done, when they're sated, then that's it. That's the end of the experiment, regardless of how interesting it is to us. And so for them, I think what's very natural about this task is not associating motion with eye movements, but effectively it's foraging. It's like trying to figure out where you're going to find your food. And they find it interesting. If you take a monkey on a very simple task, they'll peter out after an hour, maybe two at the most. But the monkeys will just continue doing this for hours and hours on end. They find it interesting. My son calls it video games circa Pong. He thinks it's pretty simple, but nevertheless, the monkeys find it interesting. Okay, so now I'd like to move to the physiology, and I'm going to show you that neurons in the brain, we have our hands, I think, on the neural basis of this decision process. Okay, so let me just explain how we think about this. Okay, effectively, I boiled a cognitive process, decision-making, down to a problem that almost seems as simple as sensory-motor integration. There's evidence in the world in the form of sensory input to the eyes, and let's just say sensory input to the parts of the brain that sense visual motion. And then that evidence has to lead to a decision, there's the question mark, of course, that ultimately instructs an eye movement. Okay, so let me tell you something about neurons in the brain that provide the evidence. We know a lot about these neurons, and it turns out we know a lot about these neurons too, so that makes our task somewhat easier. Okay, so here's the way we go about doing our work. We stick an electrode in the brain of the monkey. Here's a monkey's brain, and here's that picture I just showed you. So the idea is you take a little piece of tungsten, you coat it with glass, and you expose about 10 microns of the tip. And then you sneak up near neurons, not inside them, but next to them. And you record the extracellular field potentials from the neurons. And what that allows you to do is isolate spikes from single neurons. Now, I don't know if you stop me if I use any jargon you don't get. But the idea is that the only thing that another neuron in the brain knows about a computation performed by this neuron is the spikes that get sent down the axon. So you can think about neurons communicating via a pulse code, okay? And you can think of the spikes coming down the axon, the connection from one neuron to the next, as a point process, okay? Okay, so now I won't say much more about the form of this point process, although it's a topic I've thought a lot about. Now, I'm going to play for you what a direction-selective neuron is like. So this monkey brain you're seeing, I cut off I'm blocking you from seeing the frontal lobe. But in the back of the brain are all these areas that are marked with V's, and all those V's stand for vision. It turns out that a good fraction of the monkey's brain is devoted to vision. That's sort of true for us, too. And so let's not worry about what all these V's stand for. But here's a neuron recorded by Hubel and Wiesel. You're hearing the spikes from that neuron. And you're seeing that this neuron responds when an oriented bar of light, a little contrast contour, passes from up right to down left. They're just plotting out the receptive field, okay? So that's a direction-selective neuron. And it turns out that the direction-selective neurons that tile visual space in the primary visual cortex, V1, project predominantly to this area of the extrastriate visual cortex that's deep in this sulcus, which we've opened up here for you to see, called area MT, the middle temporal visual area, sometimes called area V5. Okay? And now it turns out that over 90% of the neurons in this structure respond to motion, just like that neuron I showed you. And here's a neuron recorded by Bill Newsom at Stanford. So there you get the idea that there's a strong background discharge. Every click you're hearing is a spike from the neuron, an action potential. And it was clear that, again, this neuron responded very strongly when the random dots were moving down and to the left, and the neuron just went back to background discharge, or maybe was a little bit suppressed when the motion was up and to the right. Okay? So that's the momentary evidence that the monkey has available to him. It's those kinds of neurons. Now, of course, the monkey has to read that evidence and say, aha, I'm going to commit that I think the motion is right or the motion's left. He has to take that information about the world and reach a decision. Were you going to say something?
Very naive question. You said there's this pulse which goes from a neuron.
Yeah.
What is additional information in this pulse? How strong it is? Or
No.
Duration?
The only thing that matters about the pulse is when it occurred. Okay, so basically in Cortex anyway, the only thing that probably matters about what one neuron communicates to another neuron is the rate at which it's producing those pulses. Okay? So you can think, if you heard the sound of those spikes sounded a bit like the clicks from a Geiger counter. And if you look at the interval distribution of spikes, it looks approximately exponential. Okay? They're highly irregular spiking patterns in the cortex. And there's a raging debate as to whether that represents a broad information bandwidth for representing lots of information, or whether or not, in fact, you should think of that the way you'd think about radioactive decay as a random point process. Okay? The latter, I think, is correct, but it's a point of controversy. Okay? So, but for the purpose of today's talk, and really for the purpose of any progress in neuroscience, in Cortex, the idea is that you should at least think about the rate at which a neuron is producing spikes. Okay? And you heard that in the video track, right? You heard that the neuron responded more. What I mean by more is a higher frequency of those pulses. And you can think of it as a point process, meaning specifically that the height of the pulse and the width of the pulse, none of that matters. It's just when the pulse occurred. It's a sequence of events in time.
I think there could be some exception for the auditory cortex, which we know that the timing somehow plays an important role.
We know that in subcortical structures in audition. We don't know that in auditory cortex, actually. Okay. We know very little about auditory cortex, sad to say. Okay, so, okay. And we could talk about that. That's an area of what is the neural code of information and so forth, and it's a topic I've thought a lot about. Why are neurons as noisy as they are, and how that might limit perception, motor control, things like that. But I'm not going to really talk about that much today, although we'll always be kind of dancing around it. Okay, so all right, so that's the sensory evidence. Let's now, the question is, how do you take that information and convert that, in effect, to an instruction to move the eyes? Well, when I was a postdoc at Stanford with Bill Newsom, what we started thinking about were structures in the brain that received input from the appropriate neurons in vision and shipped their output to the appropriate neurons in the oculomotor system, the eye movement system. And there's a lot of candidates for that. And I've studied neurons in the prefrontal cortex. Bill Newsom has studied some neurons in deeper layers of the superior colliculus. But the area that I think has paid off the most for us is this area in the parietal cortex called the lateral intraparietal area, or LIP. I'm going to call it LIP for the rest of the talk, okay? Yeah.
Block diagram up there, as simple as it is, implies some independence assumptions, which I don't know if you've actually confirmed or not. For example, might it be that the decision actually occurs in motor output as a summation, as a winner takes all, that far downstream, or is it really a separate module you think that somehow makes a decision and passes on a singular decision to the motor output?
Well, actually, I'll show you evidence, and we have direct evidence that it's the motor system that is actually doing the inquiry. Okay, and we'll get to that at the end when we talk about what I mean by an intention-based architecture of information. So absolutely, I do not mean that these are independent stages. Okay? All right, so here's what you need to know about neurons in LIP, in the lateral intraparietal area. Okay? They have what might be called spatially selective, eye movement-related, persistent activity. And what I mean by that is that if you flash a target, say, in this region of space, shown here in gray, okay, it just goes on and off, just like my green pointer. Whoops. And then you look at the neural response during a period of time where there's nothing on the screen, but the monkey's holding his gaze steady at the center here. And you'll see that the neuron discharge is elevated. Each one of these tick marks the time of a spike. And if we take the vertical averages, we get instantaneous firing rate as a function of time. Now, sometime, a random time later, we cue the monkey to make an eye movement to the remembered location of that briefly flashed spot of light. Okay? And what you find is that the response then maybe has a little bit of a buildup just before that eye movement to the remembered location of that light. And if the light is presented someplace else in some other part of the visual field, you don't see this persistently elevated activity. So the persistent elevation activity doesn't depend on the presence of a visual stimulus. It does not obligate an immediate motor response, okay? And it's spatially selective. Now, you could argue that this is related to working memory, as many people have, or directing spatial attention, or planning a movement. Okay? As far as I'm concerned, we can't sort those out, of course, from this experiment, and it's hard to sort them out in general. I think the important thing to note, though, is that this persistent activity, I think, is probably a rudimentary form, an Anlage, if you will, of higher brain function in us. And what I mean by that is that to get higher brain function, what we needed was the ability to compute and represent information in a time frame that isn't governed by immediate changes in the sensory world and doesn't obligate immediate changes in body musculature. And it's this ability to sort of separate sensation from action that allows the introduction of contingency in our behavior that allows us to higher brain function to emerge. And we can see rudimentary forms of it in simpler animals or in simple neural circuits like this one.
Okay. What part of the brain was it being seen in?
So this is area LIP, but you see this kind of activity throughout the association cortex, okay, the lateral intraparietal area. Now, and you know, these neurons care about the fact that this target that was flashed will ultimately be a location of an eye movement. Okay, if it's not, then you don't see this kind of activity. Now, and this is true of every neuron I'm going to tell you about for the rest of the talk. I'm not going to show you that many neurons. But I am going to show you in a moment a neuron recorded during the motion discrimination task when the monkey's making decisions. And you could think of something like it's really obvious that if the monkey was deciding between up left and down right, that this neuron might be informative about the decision the monkey's going to make, because the monkey's going to tell you up left by making an eye movement to a target, say, here, and he's going to tell you about down right by making an eye movement to a target here. Okay? So let me show you an example of that. So here's a recording from a neuron. You're going to see random dots in the middle of the screen. This blue thing that you're going to see is the monkey's eye position plotted at roughly millisecond resolution, although we can't have that on the video here. And then you need to know that this neuron's response field was up and to the right. So this target, we already know this target would elicit a visual response and would elicit a persistent activity before the monkey would make an eye movement there. So this neuron ought to tell us something about when the monkey thinks motion is up and to the right versus down and to the left. Now what I want you to listen, when you listen to the spikes from this neuron, I want you to ask yourself, when during the motion viewing does the neuron begin to give away the monkey's decision? Okay? There we go. A little memory period in this task. All right. So let's listen to that again. Same two trials. Already it's informative. Let's hear when it shuts off. So during the viewing of the random dots is the point I want to make. Okay, so now this neuron, in some sense, is trivial. I mean, I've told you that this neuron has a response that tells you where the monkey is planning his eye movement. And I've attached a motion signal as an instruction to make an eye movement. So in some sense, this neuron could just be telling us about the outcome of the decision process, with one hint that there might be more going on than that, which is that the modulation of the neuron's firing rate is occurring during the period that the monkey is watching the random dot motion, presumably when he's making up his mind. And so now if we go to the reaction time version of this task, we can actually see these responses evolve during the decision process. And that's what I'm going to show you on the next slide. So this is the firing rate, average firing rate from about 60 neurons in 2 monkeys, about the simplest thing you can do with data. And plotted as a function of time. And time 0 on this graph is when the random dot motion first turned on. Okay? Now this solid gold curve are trials in which strong motion instructed an eye movement to the target in the response field of the neuron, this gray region here. And the dashed gold curve are the trials where strong motion instructed an eye movement to the target away. Okay? Now, it's kind of fun to look back in time. So before the random dots come on, the neuron has an elevated firing rate. And that's not too surprising because there's a target sitting in its response field. Then the neuron undergoes this sort of dip and recovery, which we think is like a reset of a neural integrator for reasons that will be clear in a moment. But in any case, it's not very informative about the direction of the motion, right or left, or about the eye movement the monkey's going to make into the response field or away, until about this point. Okay, and then you hear this buildup of activity or this decline in activity, just like you heard in the example. Now this is the pattern of activity for a strong motion strength. Let's look at the pattern of activity when we show the monkey a weak motion strength.
I'm confused. What's the difference between T in and T out? Because in the picture they're quite.
So this gray region represents the place in space that would have elicited a sustained activity if the monkey was planning an eye movement there.
So it was there initially, was looking at it.
No. So yeah, we've screened the neuron. We already know what the neuron's where its response field is.
Oh.
Okay.
I see.
So this is a neuron that should tell us about an eye movement here, and it should suppress its activity when he makes an eye movement here. And sure enough, that's what it's doing. Okay?
It's narrowly defined goal for this.
Right. Right.
So,
That's right.
So, maybe this is no context for this though. So if you could have two sets of neurons that will respond, they would be. Why is it that they necessarily are mutually exclusive when these two sites are much more likely than anything else in the visual field, given the training and given the setup of the experiment?
Well, initially, they both look like this, right?
But are they both higher than average?
Yeah, initially, right.
They never, given the game is being played, they never found down to labile?
Well, you'll see that they do go down as the monkey is forming his decision. You haven't really seen the end of the decision process because we're cutting off the trials here. I'll explain that in a second. But in any case, yeah, they do get suppressed. So, for example, when this neuron, which cares about this eye movement, on the trials where the motion causes the monkey to make this eye movement, you see a suppression of activity, a decline in activity. Okay? I'll explain why in a second. Let's build up the rest of this graph.
So, do you believe that this kind of lateral inhibition across two, or what do you want to call it, the analog to that, is something that arises implicitly, or is actually some explicit mechanism that shuts off one alternative when the other becomes dominant?
I think that this is a way of reading out the information in the visual cortex. And so I don't know about lateral inhibition per se, but you want to look at a different signal. And I'm going to show you direct evidence that these neurons are integrating the difference in activity between two signals. And it's that difference in activity that turns out to be a useful shortcut to computing log likelihood ratios. That's basically the punchline. Okay? So let's go through the data a little bit, though, just to get a feel for what's going on here. So I think this is the critical observation, is that when you show the monkey a weak motion strength, you also see a buildup of activity before this eye movement. And you see it's hard to even call this a decline. It's called a meandering of activity before this eye movement. But you can see that the rate of buildup or the rate of decline is obviously muted. Okay? Now, same eye movements here, same eye movements in the dashed, but very different pattern of activity. If we look at an intermediate motion strength, we see an intermediate pattern. And we can fill in all the motion strengths from the easiest to the hardest, from the easiest to the hardest, and see this very nice change in activity expressed in the parietal cortex. Now, even if we show the monkey what we call the 0% coherent motion stimulus, and that is, this is just pure noise, there's no right or wrong to this, okay? Now, you see that the activity does seem to sort of distinguish what the monkey thinks the direction of motion is. And that kind of pattern also holds on error trials. This is a neuron that doesn't really say what the direction of motion is in the world. It's saying sort of what the monkey's evolving impression of that direction of motion is. That is, you could sort of think of that as the evolving evolution, the evolution of a plan to move the eyes here versus here.
Is there anything implied in that graph about the actual decision timing, or is it part of the?
Yeah, it's further downstream, so we'll get to that now. So these curves are all drawn to different lengths, and that's because this is a reaction time task. And so when we make these averages, we do two things. First of all, we clip these averages at the median reaction time so that we have on order half the trials contributing to every point or more. And we also clip out any activity in the response that is within 100 milliseconds of the actual eye movement. And the reason that's important is because when you're looking at these responses, I want you to be convinced that you're looking at the activity evolving during the time frame that the monkey is forming his decision but has not yet committed to moving his eyes. Now, all the trials, as Eric just mentioned, all the trials are going to ultimately end in an eye movement. So we can look at the responses now aligned to the eye movement. So 0 on this part of the graph represents the beginning of the eye movement response. And what you can see is that although these curves build up at different rates at the beginning of the trial, they sort of reach a very stereotype kind of level about 100, really about 70 milliseconds before the eyes begin to move. It's a little hard to see when I group the trials by reaction time, by motion strength. But if I shown you a graph of the responses sorted by reaction time, you'd see that they kind of come into a point about 70 milliseconds before the eyes begin to move. And this leads to a very simple idea about this decision process. It's as if something is building up, and when it reaches a critical value, bam, the decision process stops, and the monkey says, I have enough information, I'm moving my eyes. So this leads us to a very simple hypothesis about what this parietal region is doing. We think it's acting as a neural integrator. So if you think about it, remember I showed you that demo of the random dots causing a direction selective response in MT. The response was very noisy, but it was sort of sustained at a pretty roughly static level so long as the motion stimulus was on. All right? And so if you think about it, you could sort of characterize that as a step in activity. I'm ignoring the noise part of this for the moment. This is sort of the deterministic component of the sensory response. It comes on when the stimulus comes on. It goes off when the stimulus goes off. Now, if you take the integral of this step, you get a ramp. And if you have a stronger motion, you get a higher step. You get a ramp of higher slope. And to explain reaction time, you say, well, there's some threshold or criterion. And of course, you reach that criterion earlier if the ramp has a higher slope. So this is about the simplest idea that you could put forth to try to connect reaction time or decision time to what's going on near LIP. And it's roughly consistent with what we saw in the data slide. Now let's get a little bit more sophisticated. This kind of gives us an idea about what might be going on. I want to put a little flesh on this and go back to the behavior. And now to do that, I want to draw an analogy between a decision process and a particle diffusing between two bounds. So imagine a particle diffusing in this up-down dimension. You could sort of think of this trace as an example of the path that this particle might take. And you could sort of think of this path as the accumulation of momentary displacements, say, drawn from this Gaussian distribution. Now, I've drawn this distribution with a mean slightly above 0 in order so that we're sort of thinking about this particle as having a tendency to take steps in the positive direction. So overall, there's a drift. So this mean becomes the drift rate on average of such a random walk or diffusion. Okay? Now, by analogy, we can think of decision-making as accumulating not momentary displacements, but evidence in favor of one proposition and against another. Okay? So, and then we would imagine that we might stop the decision process when we reach this bound. If we reach this bound first, then we answer proposition 1. If we reach this bound first, we answer proposition 2. And then the reason I like this, or many people like this model, is because it allows us to, with a single model, explain both the speed and the accuracy of a decision. Now, let's relate that to the, I should mention this is not an original idea. It was first proposed by Abraham Wald in this country, and it was, this was actually the technique that Turing used to break enigma. And again, I'll get back into more details on that in a moment. And then many people have applied it to problems in perception. And most notably, I would mention, if anyone really cares to read about this stuff more, I would highly recommend a book by Stephen Link called The Wave Difference Theory of Similarity and Perception, or something like that, 1992 book. OK, now let's relate this to our particular problem. It's relatively simple. So let's say that the momentary evidence now is a difference in spike rate between, say, rightward preferring sensory neurons and leftward preferring sensory neurons. And I'll show you direct evidence for this subtraction operation in a moment. Okay? So, and we'll make the further simplifying assumption that this difference, okay, which I'm going to call mu for the mean here, is proportional to the motion strength, represented here by uppercase C. So there's one degree of freedom here, which is this constant of proportionality. Okay? Now, the only other important parameter in this model is the bound height. Now, when left and right motion are equally likely, we might as well assume that the bounds are symmetric about the starting point. So there's only one more degree of freedom. And you could think of this bound height as instantiating the speed-accuracy trade-off. In other words, the higher you make the bound, the more accumulated evidence has to accrue before you stop the process. But of course, that costs you time. Okay? So we know the nice thing about this is that there's really only 2.5 degrees of freedom here. There's the B, the bound height, and there's this constant proportionality, and there's also some non-decision time. That's really the third degree of freedom. Now, what I didn't tell you before when I showed you the behavioral data was that the smooth function through the points was actually a fit to this model. So here's the bound height B, the motion strength C. Here's that constant proportionality. And it turns out it gives a characteristic shape to this function, which is described by a hyperbolic tangent. And then we can shift this curve up and down as a unit by adding some non-decision time. Okay? And I'm assuming that non-decision time is independent from the decision time. And you can see that this does a pretty good job of fitting or capturing the overall shape of the reaction times. But what's, I think, really remarkable about this is that once I fit the reaction times, I have a value for B now and a value for K. That completely determines the accuracy. So the other curve I showed you, when I showed you accuracy, probability correct as a function of motion strength, was not a fit to the data. It was the predicted accuracy, just measuring the monkey's reaction times. Okay? And I think this is quite remarkable. You see that it misses the data points ever so slightly in various places. But I think overall, this is pretty cool. So, and so, there are problems with this model, and we have lots of fixes for some of those problems. But I think in spirit, it's a very simple model that captures a great deal about this process. And you might recognize this function as logistic, and I think there's a very deep point about the psychometric function at its heart being logistic. If you want later, I can tell you how general this idea is. Yeah.
Another question. So you have your monkey. you need to train it that if he understands the rules of the game, what he is getting food for.
Exactly.
Now, when the monkey knows the game and plays it for a long time, according to this model, if I understand you correctly, he should not become better and better, because the rest is neurons. It's not. So does it in reality? Is it the case, or if they play and play and play, they become better and better and better?
No, we only do these experiments when the monkey has achieved an asymptote in his performance. We've done this for enough years now that in other words, he's not improving anymore. Now, he's not improving anymore in fixed duration experiments where we control the motion viewing. So we think he's limited by signal-to-noise considerations in the brain. And we think we understand those signal-to-noise considerations at the level of the sensory cortex. Very important. Okay, so that the noise that's being accumulated in that diffusion process is not noise at the level of the accumulator, but noise that's coming from the visual cortex. Okay, so you're accumulating signal plus noise. Okay?
So he reaches his biological limit.
Yeah, sort of. But now let's think of another point, which is that in principle, if you had a perfect integrator and you could put the bounds anywhere you want to, then in theory he could achieve any desired level of accuracy if he was willing to wait long enough. So what we've shown, I'm not going to talk about it, is that if you assume a cost function of the form inverse reward per unit time, right? Well, let's just not call it a cost function, but just consider the reward per unit time the monkey gets and try to maximize that. That's roughly where the monkeys hang out. They place their bound at around there. With humans, what you can do is you can ask humans to change their speed accuracy trade-off, and what you find is the only thing that changes in this model is the bounds. Okay, so that constant proportionality that connects the sensory signal to effectively signal to noise, okay, I'm assuming constantly, anyway, that doesn't change. That the only thing that changes is the bound. So it's another very appealing aspect of this model. Okay?
So on the diffusion of bound model, has been probed a little bit with more complex situations like showing the stimulus half the time one way and then trying to reverse it midstream and then seeing if this equation holds?
Right. So we've done that experiment, sort of a version of that. You have to, when you're working with monkeys, or even humans for that matter, you have to say why it is, on what basis you're being rewarded. You have to be very careful with the instructions. So what we do is I'll show you a visual illusion that kind of points out what the stimulus manipulation was. So basically, we show the random dots, but in the background, instead of having black, we have kind of a very dark, kind of looks like Lake Washington today, okay? Very dark, but undulating background noise. And then we insert a little pulse of motion energy. Whoops. And then that turns out to affect the monkey's decisions later in the game. So in other words, a little pulse occurring 100 milliseconds into the motion viewing can affect choices 800 milliseconds later. And it affects the monkey's reaction time and choices according to the way you would expect from this model. And it affects the neurons, importantly, for prolonged periods of time that match the data as well. So that match the behavior observation. So in other words, that's a test of this integrator hypothesis.
But there's a fair amount of stationarity in terms of the assumed parameters, as opposed to you mess around with some odd simulation and end up with nonlinearities in parameters where this model falls apart.
Absolutely. So the monkey.
Absolutely. Which way? Absolutely.
The model will fall apart.
Which way should I gaze?
So the model will fall apart if integration is not the right strategy, and in most situations integration is not the right strategy because for a variety of reasons, let's just say, and so for the most part we don't integrate over long periods of time. In this particular instance, there's not much to be gained by kind of connecting through some, think about a belief network that would care about, say, detecting the orientation here might be detected by having a little hint of verticalness here. And then it would say, well, is there more support for verticalness elsewhere in the visual field? That's the kind of operation that you would think ought to occur in a snapshot, at least a fixation window. So you wouldn't want to integrate as a function of time. Furthermore, if you don't know that when to start integrating, then it's kind of silly. Or when signals are important in the world, then it's silly to integrate because you're just basically accumulating noise. So there's lots of situations where integration is not the right strategy. And one of them, for example, is if you say the motion could reverse at some time, and you should pay no attention to that because it's not going to get you reward. That's another situation where you don't want to be integrating flatly across time.
That's actually a high-level question then. What one wants to do to be optimal is different than what the analog to a ROM of layers of neurons will do no matter what. So the question I have is you're talking as if there is a sort of a optimal strategy selection going on to decide whether or not integration is the right thing to do or not in the situation, versus this is the reflex behavior of these layers of neurons. They will integrate. They will do X. So where do we get this idea of it's the wrong or right thing to do, and the brain figures that out? Integration versus not.
How does that happen? I mean, we have evidence that it happens, and so
in this case, we have evidence that the monkey would not integrate if it wasn't rational to integrate.
That's right. That's right. In fact, the monkey doesn't perfectly integrate to a stationary bound either. I might as well throw this idea out at you, which is that in this simple model, the bound is flat, right? And the fact is that the bound kind of collapses over time. We have some evidence that it's not really that the bound collapses, that you add a time-dependent signal. You can sort of think of that psychologically as a sense of urgency, that by a certain amount of time, you better have answered, okay? And there's good reasons for that. And this relates to some of the work we do on time perception, the fact that these neurons also represent the passage of time. Anyway.
Long discussion, basically.
Yeah. But we can come back to it. I mean, it's very, very pertinent to what I'm talking about. Okay, so anyway, this basic idea, accumulation to bound, is very similar to what we're seeing, kind of explains these data. You have to sort of extend this a little bit. So instead of thinking about a single graph where this bound represents choose right and this bound represents choose left, we really have to think about two kinds of LIP neurons, or populations of them, really. One that cares about choosing right, and another set of neurons that care about choosing left. We happen not to be recording from it here. Okay? And so then, effectively, we really have to think about a race between a set of a process that looks at right minus left, and another process that looks at left minus right. Now, because right minus left coming out of the visual cortex and left minus right coming out of the visual cortex are highly anti-correlated, we can get away with representing the process on a single graph. But to the degree that they are not perfectly correlated, and particularly to the degree that only one of these processes controls the termination of the response, we see sort of differences between these responses and these. Like, these don't look like the mirror image of these sort of across this a horizontal line. Okay? So there are slight differences. We can sort of think about this really as a race between two mechanisms. And that's important, actually, because if we want to extend this basic framework to more than two choices, we don't really want to think about diffusion in high dimensions. We want to think about races among n mechanisms. And that's work that's currently going on in the lab. Okay. So I'm just going to summarize a couple of results. I'll show you a little bit of data behind one or two of these. But in the interest of time, I'm not going to tell you about everything we've been doing. So one is I've shown you that LIP represents something like the integral dt, the accumulation of momentary motion evidence. What I forgot to say on this graph is that these averages, you can sort of think of these as the conditional averages of these kinds of trajectories. That is, this really does explain quantitatively what we see in the data. In other words, if you think of all of the motions that will ultimately end in this upper bound and take their averages, then what you'll see is the graphs that look just like this. Okay? And so that explains why even at the 0% coherent motion strength, when you're just looking at pure noise, effectively we're looking at Brownian motion. The expectation, of course, of the Brownian motion is 0, right? But of course there's these fluctuations, and you're guaranteed at some point you're going to hit this bound or this bound. So if we just pull apart and plot the average of the Brownian motion trajectories that reach the upper bound, we get responses that look like these blue curves. Yes, Eric?
We do have extra time for discussion, so I feel this is too invasive. So what do we know about the role of large populations of these guys versus small numbers of these guys having influence in the end?
Yeah, that's a good.
Obviously, these are just single neurons here.
So we're averaging across many neurons. So the assumption that electrophysiologists typically make is that it's kind of an ergodic assumption, is that when you take responses from many trials and average them, that looks like what the average of many neurons would look like in a single trial. Okay? Now, that's not true. The assumption that is false there is independence. The neurons do covary somewhat in their noise properties, and they covary on a relatively short time scale. And that actually is the source of what I think is the bound, the limit on fidelity of the neural code in the brain, is the fact that neurons in signaling as a group. Let me just give you a little riff on that real quick. Okay, so in cortex, unlike subcortical structures, you're not operating on single spikes. So someone mentioned the auditory system a little earlier, and it's very clear that a spike comes in from one ear and a spike comes out this next neuron at a very precisely timed to the first spike, contingent, say, on another spike from another ear timed appropriately, okay? And it's really the neural machinery that allows this to happen is absolutely fantastic and beautiful. There's nothing like it in cortex. Cortex is very different. Cortex is computing new quantities, combining new information in new ways, and it's computing with spike rates. And the important thing to realize that means is that if I want to add the number A and the number B, okay, and A happens to be at this moment 10 spikes per second, and B happens to be at this moment 100 spikes per second, you don't want to wait 100 milliseconds between spikes on average for this 10 spike per second signal to be valid. You want to compute in the interspike interval. Okay? The brain, needs to compute in the interspike interval. Otherwise, we're assuming unbelievably terrible bottlenecks for computational speed. So to compute in the interspike interval, you need lots of neurons. So at any moment in time, you want to be able to say, okay, in the column of neurons in cortex, I want an estimate of the spike rate. And now suddenly that exponential distribution of interspike intervals suddenly looks like an advantage, because there's no privileged times. At any moment, I have a sample of effectively spike rate. Okay?
But the point is, that's a waste of information.
It's a waste in one sense.
Out there, we know that, sound localization, the reason why that happens is because there's a correlation, right?
That's right.
20 years.
Right.
But once you go to cortex, if the information is lost.
Well, you have a different kind of information. You don't have information in sound localization. Say, the sound localization nucleus doesn't tell you anything about the intensity of the sound, say. Okay, we have an analog quantity in cortex. It's through a pulse code, but you have rate. And you can do interesting things with rate, okay? Like compute motion, for example, with it. Correlations over space and time. And so there may be interesting information conveyed in the timing of spikes, but I think actually you could make a reasonably coherent generative argument for why it is that actually the information about the timing between spikes in cortex is lost. It's lost to the history that if we imagine this is a membrane potential and this is a spike threshold and this is the reversal potential for chloride, let's say, then you could the same basic idea holds in cortex, that effectively the history of all the information about spike timing coming into the neuron is lost in the trajectory that the membrane takes to the next spike. That's an argument that I think is kind of compelling. So the idea is that it is very information lossy in that sense. On the other hand, it's extremely powerful in the sense that you can now compute in the interspike interval.
And who said that we're optimizing here, right? These are satisfied things that done well evolutionarily. So.
Yeah, so optimized for what? That's right. So optimized to be able to have a brain that does what our brain does.
Embryogenesis,
if you don't have better optimized brain, then you have the brain size will do the kind of job you want to do.
No, there's a problem there too. There's a wiring problem. Okay, the brain is, I think, a fundamental limitation in neural computation is the wiring problem, and we can talk more about that. Okay, so I know it's very disconcerting. Many people that come at neuroscience from the point of view of information theory, physics, they are very upset when I say that actually these interspike intervals are effectively like a Poisson point process. That is, there's no information carried in when the actual click from your Geiger counter occurs. All that matters is the rate at which it's occurring. And they're very upset about that because they say, my God, what a loss of information carrying capacity. But I think it's actually a very good thing. It allows neurons to compute in the interspike interval, which is not like nothing that these subcortical structures can do.
Is TCP/IP optimal?
All right. So, okay, that was a long digression. So I don't have to tell you about the evidence that LIP represents the integral, because that's this perturbation experiment where we perturb the motion ever so slightly with this little pulse of energy. Okay? And now I will tell you about some evidence that shows us that the experiments that bear on the following 2 points, that the momentary evidence is a spike rate difference from the visual cortex, area MT. And I'm also going to show you that the accumulated evidence used by the monkey really is in area LIP. Okay? So these are microstimulation experiments. They're really fun to talk about because we actually can manipulate the neurons in the brain and cause changes. Now, I should have mentioned this because you probably don't know this, but the brain itself it sounds kind of weird to say that it's not innervated, but it doesn't have any sensory neurons. So the monkey is not under any discomfort when he's doing these experiments. Anything that was uncomfortable to the monkey, we would do under surgery with anesthesia. The monkey's wide awake and happy as a clam doing this experiment. And when the electrode's in the brain, he's not aware of it. He can't feel the electrode. Now, in this case, instead of recording the activity from the neuron, what we do is we pass a tiny bit of current, like in this experiment, 5 microamps of current, for very short pulses. And the way the experiment goes is we identify now the receptive field of the neuron. So let's say this is a neuron like the one I showed you from area MT, one that would respond to random dot motion. And let's say it's direction selective and it prefers rightward motion over leftward motion. So what we do is we ask the monkey to make decisions about the motion. We put the random dot motion over the receptive field of the sensory neurons. We're out of LIP now. We're in the sensory cortex. Okay, so these are neurons that that ought to be providing the momentary evidence for the decision. Now what we do is on half the trials, we again the monkey's performing this reaction time task. But on half the trials, while the monkey's viewing the motion, we stimulate these neurons. So we stimulate with a low current. We're probably stimulating on the order of 100 to 300 neurons. But of course, all the neurons that those neurons talk to, too. So this is probably ultimately a big effect. Nevertheless, it's known that when you do this kind of experiment, you can cause the monkey to make more rightward choices. Okay? Now, what we do in this reaction time experiment is we can learn, again, we can sort of test the speed accuracy trade-off again. So what we get is if we don't stimulate the monkey, we can plot the monkey the probability that he chooses the right direction. I don't mean right as in correct. I mean right as in right and left. And this axis is a little bit different than motion strength. You see 0, the hardest motion, is sitting in the middle of the graph. And strong motion to the right, we're identifying with positive motion strengths. And strong motion to the left, we're identifying with negative motion strengths. Okay? And this is just a logistic function that says the probability of choosing right is a function of the strength of motion. Okay? So this is how the monkey distributes his choice. He always chooses right when the motion is strong to the right. He always chooses left when the monkey is strong, when the motion is strong to the left. That is, he never chooses right. And in the middle, he's distributing his choices. Okay? Same kind of graph I showed you earlier, but it's not about accuracy. It's about actually what he does with his choices. Okay, now when we stimulate, here's what happens. We shift this curve. You can sort of think of this in 2 ways. At every motion strength, you've increased the probability that the monkey will choose the direction of the stimulated neurons. Okay, we've just added some spikes into the monkey's brain, and bam, he's more impressed that the motion is to the right. And you could also think of this equivalently, based on these fits, of thinking about it as if we just added motion to the visual display. That is, we shift this curve to the left. Okay, so now, so in other words, it took for negative 15% motion, that now acts as if we had 0% motion. Okay.
Move that with current?
We've never succeeded in moving it with current, except in like really gross ways. So we don't have control of this knob, okay, which is, I think, kind of interesting. If you make the current too high, then you get something that looks more like lesion effects. So you see this curve flattening out. Okay, it's as if you're starting to activate other neurons nearby that are maybe representing the wrong direction motion.
What frequency are you?
We're using about 200 hertz biphasic stimulation.
So nothing changes as you go, like from 60 to 200?
We haven't done those manipulations in these experiments. Bill Newsom and his colleagues did them about 10 years ago, and they didn't really find big effects.
So there's like a threshold that it...
Yeah. So, we've done calculations, which I can't explain in detail, but just in the interest of time. But it's basically this kind of effect. It's as if you've affected the signal that we think the monkey's using to make his decision, where we think we're affecting like 2% of that signal. Okay, we're only affecting neurons in a small region, but the monkey's obviously, the response to the stimulus involves many more neurons than the ones we're recording.
And just another very basic question. There's still deep evidence for adjacency and locality in the brain such that neurons reside near each other that are doing sort of the same thing, having the same kind of sensory effects, for example, in this case.
That's right. But we wouldn't do the experiment. We couldn't do we choose to do the experiment only because of that, and that's a condition for actually choosing a site. We want to make sure that all the neurons near the tip of our recording electrode seem to share the same selectivity for motion.
I mean, I'm just amazed that that's often an implicit assumption for the whole cortex. And it didn't have to be that way whatsoever.
It didn't have to be, but if you look, again, consistent with this idea about wanting to compute the interspike interval, if you have one neuron, what you're saying is that if I want to communicate to you some spike rate, I'm telling you that you better listen to me and my brothers and sisters, okay? And so, and otherwise, you can't know spike rate. In order to know that, we have to share wires. In order to share wires...
Yes. Let's say if wires were really cheap and inexpensive,
that's right.
Space was abounded, it could have been scattered throughout the whole brain diffusely.
That's right. And that's sort of the myth of the neural net, okay, is that you can connect anything to anything. But the real architecture of the brain is that you can't connect everything to everything, okay? And that's a really important... That's why I said that connections and wiring length considerations are really important computational principles that we ought to not be ignoring. Yeah.
I wonder if you could also were able to shift it to the right by like numbing or turning off.
We've never done that.
Okay.
Yeah. So right. That's a bit of a blunter. Oddly enough, that's a blunter instrument, and it's one that we invest so much work in getting these monkeys trained. The last thing we want to do is do anything that would actually hurt the brain that we're recording from. But we're going to do those experiments, actually. Those are on the books. OK, so this is an old result, actually. This is just a confirmation of Bill Newsom and Dan Salzman result from about over 10 years ago now for choice. What I want to now show you is what happens to the reaction times. OK? And that's much more interesting. I'm going to build this graph gradually. So first, let's look at when motion was to the right, OK? And the monkey chooses right. Then you see again that the reaction times shorten as we make the motion stronger. And when we stimulate and cause the monkey to make more rightward choices, we also speed up his reaction time. Okay? Now, here's the more interesting case. So when motion is to the left, and the monkey chooses left, once again, if we make the motion stronger, the reaction times go down. But now, before I put this graph up, I want you to think about what's going to come. So we're stimulating rightward neurons. And I'm going to show you trials where the monkey effectively ignored the stimulation. He chose leftward. We stimulated the rightward neurons, but we failed to influence the monkey's choice. But what happens to the reaction time is that it slows down. Okay? So what this tells us is that the rightward preferring neurons in the sensory cortex are influencing leftward decisions. And if we looked at this, if we actually fit this more quantitatively, I would convince you that the change is subtractive. Okay? That it's as if the rightward neurons, when they increase their discharge, are providing negative influence on leftward choices and positive influence on rightward choices. Okay? Okay, so that's the direct evidence for two things, that MT neurons are in the causal chain of this decision process, and that the decision... Whoops. I don't think I did that. Okay, so... Ooh, that's very bad. Okay. That MT neurons are in the causal chain, and furthermore, that the way that they are read out is by seeing a difference, is by subtracting the two's firing rates. Okay.
Is there ever a paradigm in this or a sub-paradigm where you actually only allow response at a certain time? You peg the response time, you anchor that, and what happens to accuracy, the simulation, stimulus?
We have not. We have not done this experiment. Well, I'm not sure exactly. So you're saying you must respond by such and such a time?
He knows that no matter when it thinks it has its decision, it can only answer at 800 milliseconds.
Yeah, when we do that experiment, then of course we can no longer measure reaction time,
right.
But we still get effects on the choices. Okay.
But you can require it at 400 milliseconds or 200 milliseconds too.
Right.
Okay.
So we actually have never done that experiment where we varied the duration in this particular protocol, but we've done enough other things that would be
okay.
like that. Okay, so I think I'll skip this experiment just to say that when we stimulate an area LIP, now of course we're stimulating neurons that no longer represent the motion, but effectively represent one of the choices, right? This is these neurons that have this persistent activity before choosing this target or this target. And when we stimulate those neurons, what we find is kind of a similar effect, smaller effects, speeding up reaction time this way, slowing it down for the other choices. It's very similar to what we got in the sensory cortex. But what we find is that the effect on the actual choices, it's there, but it's much smaller than what I showed you for the visual cortex. And just to summarize these effects, it turns out that that when you stimulate area MT, it's as if you added a spike rate to the momentary evidence. And therefore, what happens in the accumulation is that you get an accumulation with a higher slope, okay, a higher drift rate. Okay? It's as if, basically, it's as if you just showed a stronger motion strength on the screen. When you stimulate an area LIP, what I've just told you is that you get more right choices, but it's a tiny effect. And again, you get faster right choices and slower left choices. Okay? But that turns out the pattern of activity, choice, and accuracy is not as if you've added to the momentary evidence, as if you left that alone, but rather you just offset this accumulation. You just added a number to it. Okay? And that has a more profound effect on the reaction times than it does on the choices. It's effectively quadratic in reaction time and linear on choice. Okay? And so basically, we're very excited about this because we think we have our hands on 2 points in the system that are in the causal chain of a decision, and they have different kinds of computational roles in the decision process. One is representing the evidence, the other is representing the accumulated evidence, really a decision variable that's being compared to some criterion or bound. Okay. So I just, I should tell you there are things we don't know very little about. We don't know how the integral is actually computed. Okay? We don't know even where it's computed. We know that LIP represents the integral, but we don't really know that LIP is the site where it's being computed. And we don't really know how the bound is set. I've already alluded to the idea that the bound establishes the trade-off between speed and accuracy, but we don't know how it's set, and we don't know how it's read out. Okay.
If anybody would change the delicious quality of the reward and see that if the trade automatically values somehow.
People are beginning to do those experiments where they play off reward against choice, but they haven't done that. Bill Newsom is actually doing that experiment in a fixed duration version of this. So you can't look at reaction time, and on our view, the reaction time is going to be diagnostic of what's going on because we expect that the reward is going to affect bound height and should have very different kinds of effects on reaction time. And I don't know whether we'll have time for it, but when we manipulate prior probability, like make rightward more likely than leftward before the motion ever gets turned on, we can see what happens to the responses as well. And that's a related manipulation. What happens is not what you would expect from Bayesian thinking. What happens is that the prior probability is instantiated as a change in momentary evidence. It's as if instead of acting as a criterion on the evidence, it acts in a way to basically perturb the evidence itself. It's as if the monkey expects rightward, you've added a few spikes per second to the rightward preferring motion stimuli. It's a crazy idea from one point of view. Just the way I like to think about it is you're a radiologist, you're reading mammograms, you decide some are positive and some are negative. Okay, so now you move to another part of the country where breast cancer incidence is much higher. So what do? Well, you change your criterion, right? You don't go back and sort of like doctor up the mammograms and add calcifications and stuff. But what the brain does is it allows prior probability to masquerade as stimulus effect. And the reason why, I'll just explain in a nutshell, is because there's uncertainty about uncertainty. Because you don't really know in the world what kinds of evidence you're going to get. And so therefore, there's really no sensible way to add prior probabilities to the evidence you're getting using the right units. You don't know how to scale those units appropriately. Maybe let's spend some time on that. I think it's really kind of intriguing and a very, very clear story in human and monkey. So I've already told you about sort of the main experimental results that I wanted to summarize for you. That decisions in this very simple task are based on accumulation of evidence over time, and that the momentary evidence is a difference, and that neurons in the parietal cortex are representing an integral. Okay? Now I want to go back to Turing and tell you why this is all sensible. Okay? And it's a very Bayesian framework that I'm going to propose for you. So this is from the point of view of sequential analysis. This is a fun little paper by Jack Good, who is a well-known Bayesian, and we've summarized it in a paper of our own. And basically, it works like this. So Turing was trying to figure out, they were trying to break the Enigma code, right? And so most of breaking the Enigma code was a lot of dirty tricks and stealing machines and stuff like that. But there was one part of it that was kind of cool, which was that what they would do is they would intercept two messages, say this blue one and this green one. And they were created by two different Enigma machines, okay? And they mean completely different things, presumably. But what they wanted to figure out was, were these two messages created by Enigma machines that had the rotors in exactly the same state? That's the hypothesis they were asking, okay? So the hypothesis, messages are encrypted by Enigma devices in the same state. And if they could figure that out, then it turns out they knew something about the message during the day for the settings of the rotors themselves, okay? So what Turing figured out, what he ought to do is is he ought to do the following. He would line up these two messages, and he'd look for matches, like this E and this E. Okay? And then what he said is if you get a match, okay, like this E and this E, what are the odds of that? Well, if the machines are in random states, it's 1 in 26. And if the machines are in the same state, then it's 1 in 13, because some letters are more common than other letters, and they're just going to show up as matches because of that. And so what Turing said is, okay, take this is an odds ratio or a likelihood ratio, take the log of it, he used log 10 and multiply by 10. And he called this unit of evidence, weight of evidence, a deciband. Okay? Ban was short for Banbury, which was the town that they printed these messages on. So he called this process Banburismus. Okay? And he speculated at the time that 1 deciband, maybe 2, was the smallest amount of weight of evidence that ought to be of any use to any information, any decision maker. And he thought maybe one day this would have some impact. But of course, his work wasn't published. It was classified until the mid-'70s when Jack Good wrote about it. And so Turing never got to witness its instantiation in the brain. But I'm going to show you that something like this goes on in the brain. So now, of course, most of the time you get a non-match, like this K and this E or this C and this N. So what are the odds of that? Well, it's 25 out of 26 times if the machines are in random states, but only 12 and 13 if the machines were in the same state. Okay? And so if you crank through this arithmetic, what you get is negative 0.17 decibands in favor of the hypothesis. In other words, this is weak evidence against the hypothesis that the machines were in the same state. And so what Turing said was he only had so many, they had tons of these codes, of course, coming through, and they even wanted to change their registration and stuff. So they only had so much time to do this. So he said, well, we need a method for deciding when we're done with our decision process, to terminate. So what he says, okay, so just start gathering the evidence. Every time you see a match, bump up by 3, and every time there's a non-match, decline by 0.17. And then when you reach one of these two bounds, stop the process. If you reach this one, you've stopped saying the hypothesis is supported. You reach this one first, you stop saying, no, the machines were in random states. Okay? And it turns out that this, what I've described to you, is known formally as the sequential probability ratio test of Abraham Wald. And it's actually the most efficient test for deciding between two hypotheses. That is, once you've decided on what your type 1 and type 2 error rates are going to be, you set your bounds, and this is the best test you can do in the sense that it's the smallest sample size needed. Okay? Here, sample size, of course, is a random number, but so the expectation of the number of samples is the smallest possible to achieve the power that you desire. Okay? And the proof of that is in Wald and Wolfowitz. This is the Wolfowitz who's the father of the bad Wolfowitz who's ruining the world. Right? So and now he gets to ruin the economy. So okay. So
just the famous one.
What?
Not bad, just the famous one.
Yeah. Well, Jacob Wolfowitz was also pretty famous, at least in statistics circles. So.
So they're both infamous. We'll just leave it at that.
Yeah. Jacob Wolfowitz, I heard the big bad Wolfowitz interviewed on NPR, and he said that his father didn't approve of his politics. So I was very pleased to hear that. So okay. So now let's go back to the brain. So let's think about, so the idea behind Wald and Turing was that what you should accumulate, the critical thing was you should accumulate a log likelihood ratio. Okay? Now, so let's think about how this occurs in the brain. Well, so let's say this is the response distribution. Think of this as a probability density function of the responses we might get from a rightward preferring empty neuron. Remember, this is just a sensory neuron. It might spit out 29 spikes per second on average, sometimes more, sometimes less. This is pretty realistic. When rightward motion is shown, okay, weak rightward motion, okay, something hard, but still it's to the right. And this might be the responses that a leftward preferring MT neuron would give to weak rightward motion. Okay, so this means greater than this, which is the direction selectivity that we're talking about. Okay, there's overlap because we're talking about a difficult stimulus where errors might be made. Now, if you wanted to compute likelihood ratios, you could do things like, if you happen to get 29 spikes per second, you could look at the height here and look at the height here, and you could play the game and calculate through likelihoods and take their logs and so forth. But it turns out, since you have these two kind of populations of neurons, you might as well take the difference in spike rate between them. And if you do, then now this is the difference in spike rates, right minus left, again, in association with weak rightward motion. So most of the time the responses are greater than 0, and that's a support for the hypothesis rightward. And sometimes the responses are less than 0. That would be support for the hypothesis leftward. Now, it turns out, and this is, I think, a reasonably general statement that it doesn't depend on Gaussian assumptions or anything like that. It turns out that this difference in spike rate, which has unit spikes per second, it's very simple for the brain to compute, presumably, is proportional to a log of a likelihood ratio. So this is kind of a heuristic, a neural heuristic. It's a shortcut for doing Bayesian approximation, or for, in this case, just simple Bayesian decisions. It says instead of computing likelihoods and taking their logs, you just complete a difference, skip the likelihood step, and go straight to a quantity that's proportional to the log likelihood ratio. And just to give you a quick intuition for it, if this is the difference right minus left under rightward, this would be the difference right minus left under leftward. You can see that at least at the 0 point, okay, that's, of course, providing, if the rightward neurons and leftward neurons are providing the same response, then there's no evidence in favor of rightward. At least it conforms to your intuition at that point. And so it turns out that if you just look at the difference and plot a log of the likelihood ratio, you get a line. You get that under a variety of assumptions about distributions. And there's something very general, I could say, if you want, about a special root of the moment-generating function of distributions that make this true in a really wide variety of cases.
What's that?
It's that the number that you really want to be accumulating, okay, is if you think of a difference distribution, one that can take on positive and negative values, and you write down its moment-generating function, it will have a value. Of course, it has the value 1 at 0, okay, but it'll have another value 1, and at that root, that special root, okay, that value turns out to be turns out to be the quantity that is effectively proportional to the log likelihood ratio.
And it's still easy to compute B differences.
Yeah, you need differences, otherwise you're not guaranteed to get that root. Okay, you need a probability distribution that has positive mass in the positive and negative numbers, basically. Okay. Now that doesn't say that there's easy ways to work with that particular number in the Gaussian difference of Gaussian, different Poisson, different of gammas, different almost any distribution that we would want to talk about. It's a piece of cake. Okay, but it's more general than it seems.
My understanding about what sequential detection theory is that given the miss probability and the detection probability, you can actually optimal, according to
That's right.
theory, set up the bound.
That's right.
And that will give you minimum number of observations.
That's right.
To reach that bound. So from this neurophysiological study here, do you try to say that these bounds are close to what the optimal numbers?
Well, you have to define what is meant by optimal. So can I use the whiteboard?
Yeah.
So
that could be easily tested whether it's optimal or not.
yeah. So let me make a quick graph. So let's worry about the bound height. Let's assume equal priors and so symmetric bounds. And let's plot as a function of bound height. Let's plot the probability correct on average. Okay? And don't forget, some stimuli are easy, some stimuli are hard. So we're going to get a monotonically increasing function. Okay? Okay, there's no particularly easy way to describe this function. It depends on the mixture of stimuli that I'm using. Because some are really hard. I can put the bound height high. I'm still going to perform badly. Okay? Now, let's also plot the average time spent. Okay? Okay, obviously, again, if the bound height is high, an increasing function. Okay? Right? Now, let's plot the reward rate. This is a function that has a maximum. Okay? Now, if you solve for that maximum, you find a bound height that's about where the monkeys are willing to work. We have not succeeded in changing the bound height for the monkeys. And we think that's because we cause them to work near a natural optimum. But that's pure conjecture on our part. But it is interesting. You have to be very careful to talk about what you mean by optimality all the time, right? So in this situation, we have a mixture of stimuli. And the assumption is that we have to apply the same bound height regardless of the stimulus. So we can't do walled, you see. We can't take a stimulus and convert it to a log likelihood ratio and accumulate. If we could, in principle, we could get a flat psychometric function. Right? Because if we could compute, if we knew how to convert an extra spike from the rightward preferring neuron and could convert that to a log likelihood ratio, then even though that's kind of crap evidence when the motion's weak and fantastic evidence when the motion's strong, we could just accumulate more when the motion's weak and perform arbitrarily well.
So this reaction time is equivalent to the number of measurement that's required to reach.
That's the way to think about it. Right.
What's the concept of a false alarm in world theory?
So, yeah, well, in our case, since it's two alternatives, we can think of just whether or not you want to. You can think of false alarms and hits, to use the signal detection theory. You can think of that instead of in a detection experiment, you can think about the number of times that rightward motion produces a rightward answer versus rightward motion produces a leftward answer, or leftward motion produces a rightward answer. There's a symmetry argument at play.
So how, in this analysis here, how did you compute reward?
So there's a total number of total time is the time between the trials. Any other penalty time that we give, we don't give the monkey any punishment for getting things wrong. We do insert a timeout period, okay, a second, you know.
So you basically
wait. What?
You have to just wait.
You have to wait, right? So they want to play the game, and so there's your reward per unit time is just add up all the time in the trials based on what you'd expect this model to produce. It's a simple computation, and it has a maximum. Okay. But as I say, there's some testable predictions that we've never succeeded in supporting this. So this is conjecture. I mean, you know, take it for what it's worth. We also know that this cost function, or the inverse of a cost function, this gain function, which is reward per unit time, doesn't explain the monkeys and the humans' behavior when we manipulate the prior probability. Okay. More, I guess.
Even given Tversky's assumptions.
Well, it turns out that
Just talking about that, by the way.
Okay, so I think what I should do at this point is I was going to tell you about let me just fill in this graph just so you get an idea. So I'm saying in mechanistic terms, the brain is making decisions by accumulating something simple, difference in spike rates, right minus left. It has a neat interpretation of that as if you're accumulating something that's proportional to log likelihood ratio, but you don't know the quantity proportionality because there's an uncertain world out there, and when you start looking at it, you don't know whether you're being given an easy or a hard stimulus. And I think it's a really important consideration that makes a lot of Bayesian computations hard, where we assume we know things like likelihoods. And then you could also think of this in Turing's terms, as if you've accumulated weight of evidence, or his terms, decibands, and belief. And in a way, this is kind of related to subjective probability. It's a poor man's estimate on a posterior probability. Okay, and in fact, the fact that you get a logistic psychometric function kind of suggests that in a way the psychometric function is representing the posterior probability of making a choice. It's a little bit funny thing to say, because the fact is that this mechanism leads to a particular choice. It doesn't lead to a probability, that would parameterize a flip of an unfair coin. It leads to the choice, and that's a really important point that I think we also tend to gloss over as Bayesians. I'm a Bayesian. Okay, so I'm going to skip this. We actually tested the idea that neurons in parietal cortex represent log likelihood ratio by training the monkeys to play poker. And all I can say is that, yes, the neurons represent log likelihood ratio. So I'm skipping this. And I just want to sort of end on a more architectural point. Okay, so as Eric mentioned, I'm a neurologist, so I do actually care about the health implications. But for the purposes of Microsoft, we might also want to think about computing architectures, and I think that there's some interesting implications here. So, and really, one of the reasons I sought out this dialogue. So, okay, so simple brains channel sensory information into motor structures. Okay. And more complicated brains allow processes to intervene between them, add contingency to this sensory to motor transformation. And a decision process is just an example of one of those. Okay? Now, a decision process means that we kind of have to gather information from a variety of sources, not just the evidence, but as we mentioned, prior knowledge, expected payoff, and I think time belongs in this diagram too, urgency. Okay. And in our case, instead of just thinking about a motor output, we can think about a potential behavioral plan. Okay, not something that actually moves the body. Okay.
Can I ask you a quick question before you generalize? In a simple case, the place where evidence is integrated and where the motor function, they should be very close, right? Because they somehow the motor function somehow should quickly get this information.
That's right.
So they should be closed, right? Because integration cannot stand for a long time.
Right. Well, this is why we're able to make progress, is that we associate motion with a particular eye movement, so we can record from neurons that care about the eye movement. And it didn't have to be this way, but they end up showing us the formation of the decision. Okay. Now, another version of the way the brain could have been built, and I think the cognitive psychologist's conception of the way the brain is built is that there's a concept of rightwardness that emerges from watching the stimulus. And that concept of rightwardness need not have any relationship between moving my eyes to answer rightward or pushing a button or using a verbal output. I think it's to the right. But what I'm saying is that the brain has effectively, it's just sort of built up a little bit from a sensory to motor a reflex, really. You can sort of see the emergence of higher brain function. It's just adding a little contingency between them. And sure enough, structures in the brain that care about how you're going to answer the question are accumulating the evidence that lead to that answer. Okay? And we can break that by making tasks where you cannot associate motion with eye movements, and we've done those kinds of experiments. But when you don't, the brain assembles information in this very intentional affordance, to use the Gibson term, kind of way. Okay? And I think it's important to think of it this way. So in our particular task, we can sort of think about the association cortex, area LIP, as taking information from the visual cortex and construing it as evidence for or against a particular action. So motion up here, rightward says, oh, make this eye movement to this region of space. Leftward means don't. That's through training, of course. Okay, so there's something learned here. But the point is that the operation on the stream of information is already directed into trying to channel that information into a set of hypotheses. And that's what I mean by an intentional-based architecture. I think it's extremely powerful because a lot of the problems that we think about when we make calculations on sensory information involve computing posteriors on very high-dimensional descriptions of data. Okay, I'm thinking sensory data. You might be thinking about other kinds of data. Okay, but what I'm saying is that what the brain does is that it starts from the idea that there's certain kinds of hypotheses that are worth addressing in the first place, and effectively they're embodied. There are things like, shall I move my eyes here? Shall I reach here? Shall I grab this way? If I put my hand this way, is it likely to slip through? If I move this part of the world, is there another part attached to it that might also move? In other words, there are questions about the things in my world, and I query my sensory maps, my sensory data, for evidence for and against those kinds of hypotheses. There's a limited repertoire of those hypotheses that we entertain at any moment, and there's a limited repertoire of those hypotheses that we can entertain because we are ultimately connected to bodies. Okay? And that's a really important point. And it makes a lot of computation a lot easier, I think, by taming high-dimensional problems, or what's often referred to as a combinatorial explosion of of information. Okay? I think that's a point that actually is worth us thinking about, or worth you guys thinking about, from the point of view of even developing algorithms.
So these are hardwired, primitive level concepts, and
some things are hardwired and some things aren't. So we can sidestep the hardwiring with learning. Clearly, the monkeys are not hardwired to think that motion at some part of the visual field of a bunch of dots means move your eyes from here to here. Okay, so that's learned, and we know that. Okay, but in general, I think that there must be some flexibility in the brain in order to do this kind of routing, to know which question to ask in the first place, which is I've sidestepped that, and then to know how to go back and find information in the sensory cortex that's relevant to that, and now construe it in some way like a log likelihood ratio within a constant proportionality.
So can you talk a little bit about asking the question versus the LIPs presenting the information? What do you mean by asking the question?
So the monkey sits and he's in a context of a chair, and he knows that he's in a situation where eye movements are going to get him reward, and he's got to do this in association with motion. Okay? So the question is before him. He's learned the context, and he thinks he's basically this very, very simple decision. He says, okay, I'm going to now ask the visual cortex for evidence. I'm going to construe activity in the appropriate parts of the visual cortex as evidence for or against these particular, very circumscribed actions that I can do later into my world, make an eye movement.
But do you mean that conceptually, or do you actually see signals coming from the motor system into the LIP? Or?
I mean it conceptually, but what I've said doesn't require any kind of feedback. It requires some higher level control over which circuits are going to be active. So like the prefrontal cortex, let's imagine, controls the context and tells LIP, the right circuits in LIP, that it's time to now plan eye movements based on motion information. Okay? So there's a kind of a feedback, a top-down influence on LIP. But the rest of everything I've said is all feed-forward. You've had this by learning, presumably. You've got right minus left neurons up here, up minus down here, and northeast versus southwest neurons from this part of the visual field, all sort of projecting to the LIP neuron that cares about telling the monkey to move his eyes to here versus someplace else. Okay? And that wiring must be achieved through learning.
But you think that it doesn't occur continually? It's only when it gets in a certain context state that...
Yeah. So I think we carry around two illusions of in processing. One that we're all familiar with, that we carry the illusion of foveal acuity throughout the visual field, even though it's not true. I mean, most of the information coming through and even registering the visual cortex we're not aware of, right? I assume you've seen change blindness demonstrations. That basically a lot of the information in front of us we're not aware of. Oh, maybe I'll show you that if you like. But in any case, that's true. Okay? And we certainly don't have foveal acuity throughout the visual world. If you ask what is the panorama of vision in front of you, you will say that you have this very fleshed out painting in front of you, right? And similarly, on the motor side, we carry around an illusion of sort of pluripotentiality. That is, we think that at any moment that anything is possible. It's probably not true. Our brains at any moment when we're awake are engaged in one of n possible hypotheses or tasks, is the way I think about it. Now, what sets up which tasks when? I mean, that's a really interesting question. But the point is that there's a limit that there are constraints imposed on the processing of the data.
But pretty remarkable for some of the functions like balance, how well they recover to surprising destabilizations. The whole appropriation of the system, musculoskeletal system, how someone walking and slips or hits a rock. That one over n is probably not there, but it's probably hardwired. Being balanced and the balancing pole problem is probably really hardwired down deep for quick responses in general.
Sure.
This speculation. It's already 3:02, and I want to just make sure that given our TV studios and our massive audiences out there in TV land, we stop so people can go on to the next event. So why don't we thank our speakers and take it offline. And I guess you have a comment here about acknowledgments.
Yeah, I think I should tell you who did the work that I've been talking about. There's a ton of people, not all of them still in the lab, but I should just mention that the most of the physiology that I showed were done by Jamie Reitman and this guy, Mark Mazurek, and the stimulation experiments by this guy, Jochen Dietrich.
Being in America, I'm sure it's more than 1 ton.
What?
I'm sure it's more than 1 ton.
More than
1 ton, what do you mean?
Just weight. 1 ton is 1,000 kilograms.
Well, there you go.
It's about.
Okay, thanks very much.
A pleasure. Thank you.
Just a joke.